Hello, everyone. Welcome. I'm coming to you recorded live from my hotel room in Atlanta because I'm away for the teachers conference for a couple of days. So if you hear some background noise, some whatever, this is a hotel is downtown, and there's a little bit of noise outside. So I apologize for that up front. Let's get started. I hope you have your cell transport notes handout ready to go. So let's go. Let's talk about how cells get things inside and outside themselves. All right. So let's start off by talking about the cell membrane. Now, you've been over the organelles a little bit, so you know that one of the organelles is called a membrane. All cells have a membrane, and the membranes all have the same basic makeup or structure. They're made of lipids, specifically phospholipids. We'll get to that in a second. And proteins. And the proteins within the membrane do lots of different jobs. So here's a sort of a schematic diagram of what a cell membrane looks like. With an animal cell here and a plant cell here. Remember, they're generally the same and their membranes are the same, but with a plant cell you also have the cell wall around the outside of the cell. Okay, so what is the function of a cell membrane? Well, it basically is a barrier. It separates the inside of the cell from the rest of the world. But it's more than just a wall. It's a gatekeeper. It's an active participant in figuring out what can come in or out of the cell. So we say it regulates the flow of materials into and out of the cell. And there's a term for that, which you will see here and you will see again. It's called, there's a couple ways to say it. You can say the membrane is selectively permeable or that it has selective permeability. Either way, the selective part means choosing, so letting some things come in and others not. And permeable means allow to cross or to let through. So selectively permeable means letting some materials get in or out of the cell and preventing other things from getting in or out of the cell. All right, so a membrane is described as selectively permeable. The membrane helps the cell maintain what we call homeostasis. Homeo means same. If you think back to, we're going to think back a lot to our language of science worksheet, so long ago. Homeo means same, and stasis refers to the situation or the condition. So homeostasis means the same condition, a stable, in this context, a stable internal balance or an internal environment inside the cell. The cell has to maintain its internal environment at the optimal conditions. And finally, what I want to say is the structure of the membrane is called a lipid bilayer. Bi means two, so two layers. You can see it a little bit in this diagram, but it's actually better in the next diagram. We call it a lipid bilayer because there are two layers of lipids. So here is one layer, and here is the other layer, and that's why we call it a lipid bilayer. What you'll also see in this diagram are that there are proteins in the membrane, as I mentioned already, and there are different kinds of proteins. Some, like this protein, are they're integral membrane proteins. They go all the way through the membrane from one side to the other. And so does this one. Some of them sit in the membrane but don't actually go all the way across it, like this one. What we don't show in this diagram, or maybe that's right here, some proteins kind of sit on the outside of the membrane but are stuck to it. Other things that appear in the membrane, these green lines are supposed to represent carbohydrates, so sugar molecules. Cells have sugar molecules attached to some of their, actually not the green line, I'm sorry, the yellow dots. Are sugar molecules that are used for identification. So the cell membrane is a complicated dynamic structure. Okay, so let's take apart a little bit what those pieces are. Lipids or phospholipids is the first group. The correct name actually for these components of the membrane is a phospholipid, and that is what these blue dots with the little red tails hanging off. That's a phospholipid. Here it is represented over here one more time. A phospholipid has two parts to it, a phosphate group and a lipid tail. The phosphate group is the ball at the top, and the lipid group, there are usually two lipid tails that are attached to that ball that are the lipid part. So phospholipid. Yeah. Now, phosphates and lipid groups have different characteristics. So a phosphate group is a charged molecule. It has a negative charge on it, and that means that it's polar. It likes water. It likes to be around water because water is also charged, and so these things are happy together. The lipid tail part does not have any charge on it. It doesn't like positive or negative charges. It does not like water, and so it's called hydrophobic. Hydro means water, and phobic means fearing. So hydrophobic is water fearing, and hydrophilic is water loving. Hydro meaning water, philic meaning loving. All right. So this phospholipid molecule is kind of interesting because it has a hydrophilic part and a hydrophobic part. Now, if you put a whole bunch of these phospholipid molecules together, they will assemble themselves into a structure like this, where all the hydrophilic parts, the little balls, are next to each other, and all the hydrophobic tails are associated with each other, keeping the water away. So if the hydrophobic tails arrange themselves in this way, like this, that means that they can keep out the water and just be near other hydrophobic tails like themselves. And the head groups, the phospho part of the phospholipid does the same thing. They'll associate with each other and be near water and be happy. So that's what makes up a lipid bilayer. That's what makes up a cell membrane, structurally speaking. All right. Here's another view as if you were looking at it from the side. So here is one, maybe this is the inside of the cell, and this is the outside of the cell. And so your polar hydrophobic phosphate groups are out here, and the hydrophobic tails are in the center here, hanging out with each other and keeping the water away. Oh, just in case you don't know this word aqueous just means watery. So an aqueous environment means a watery environment, just like the one you find inside a cell. All right. So we said that one of the components of cell membrane was, of course, the phospholipid, and another component was a protein. There are lots of different kinds of proteins that you find in a cell membrane, but what we're going to talk about today are transport proteins. And as you might guess, transport proteins move things or allow things to move. So transport proteins are found inside cell membranes, so this is a transport protein. They can also be called carrier proteins or channel proteins, and we'll get to the difference in those in a couple of minutes. The purpose of a transport protein, either carrier or channel, is to allow large substances to move across the membrane. All right? You know what? This word plasma is interchangeable with cell membrane. I thought I took all of these terms out of the presentation and made all the plasmas into cells just to be consistent, but apparently I missed one. Plasma membrane is really cell membrane. All right, another component that we have, and we'll get back to the transport proteins in a minute. Another component you find in the membrane is a cholesterol molecule, and they're a little hard to see, but if you look here in the hydrophobic portion of the lipid bilayer, you'll see these darker yellow little fragments. These are cholesterol molecules. Now, you've heard of cholesterol before, I assume. We talk about having high cholesterol, and maybe cholesterol is bad for your heart and things like that. But actually, cholesterol also, and that's true, can be. Too much cholesterol can be bad for you. But cholesterol also has a normal function in your cell membranes, and its function is to prevent these lipid tails from sticking to each other. So if you see in this membrane diagram we have here, there's a bunch of these yellow cholesterol molecules, and cholesterol is also a hydrophobic molecule, so it's basically hiding from the water inside the lipid bilayer, and it also provides sort of a lubricant. It keeps the lipid tails from sticking to each other. All right, so that's what cholesterol does in the cell membrane. Okay, so let's talk about transport, because that's what this is really about. We're going to consider transport in two ways. There are two ways to look at it. There's passive transport, and there is active transport. Let's start with passive transport. The thing to remember to distinguish them is that passive transport is free. It doesn't cost the cell any energy to use passive transport. All right. So the definition of passive transport is the movement of material. It can be food particles or ions or building materials for the cell down a concentration gradient. And it requires no energy from the cell. What do I mean when I say concentration gradient? Because you might not have come across that before. So let's take a look at this little diagram, this little animation. You'll notice with these purple dots, they all start out in the lower left corner here, and they spread out across this square. So at the start of the animation, when all the dots are in the corner, we would say that there is a concentration gradient of purple dots, with lots of purple dots in the lower left corner and basically no purple dots in the upper right corner. That is a concentration gradient. Let's see it one more time. So lots of purple dots down here, not so many purple dots up here. Now what happens is, of course, you've probably encountered this before. Let's say I was standing in the classroom, and in the corner of the classroom over by the chemical hood, where the computer card is, and let's say I opened a bottle of ammonia. Ammonia smells bad, right? You may recognize the smell of ammonia from home or whatever. If I opened a bottle of ammonia in the classroom, you would say that there's a concentration of gradient of ammonia molecules where there's a whole bunch of them right next to me where the bottle is, and there are none at all over on the opposite side of the room. But of course, those ammonia molecules will spread out, diffuse away from the open bottle until, just like these purple dots did, until the whole room smells like ammonia. All right. But before that end point, we would say that there's a concentration gradient of ammonia molecules where there are lots of them in the corner of the room and none of them on the opposite side. All right. And that's what these purple dots are representing. So that has relevance for transport in cells. Specifically, when we're talking about in the context of cells and cell biology, we use the term diffusion. So diffusion is the movement of molecules from an area where there are lots of those molecules, a high concentration, to an area where there's a low concentration. That's the purple dots in the previous slide. But we have another way to look at it here. Let's say we have this beaker of water here, and we drop in a single drop of red ink. All right? And at first, all the red ink molecules are right next to each other, and so there's a high concentration of red ink molecules here, but here there's no red ink molecules. So we have a concentration gradient of these red ink molecules, and that's how it looks here. And then over time, those red ink molecules will spread out until the whole container is basically filled with red ink molecules like this. Okay? So that is the process of diffusion. The movement of something, red ink molecules in this case, purple dots on the previous page, from an area of high concentration, where there is a lot of it, to an area of low concentration, where there's little or none. And that's what diffusion means. Diffusion is actually a really important process in biology. Lots of things move in or out of a cell, in or out of your lungs, for example, gases, by diffusion. And then we have one specific kind of diffusion that we need to mention, and that's called osmosis. And it's special because it's still just diffusion, but specifically it's the diffusion of water molecules. All right, so osmosis is the movement of water from a place where there's more of it to the place where there's less of it. You're like, wait a minute, what do you mean more water, less water? Well, let's take a look at my little diagram here. So here is a U-shaped glass tube, all right? And it's got water in it, as you can see. And there are also some purple dots. And then, importantly, there is a thin membrane right here. It's a selectively permeable membrane. Remember what that means? So this membrane will let water molecules cross, but it will not let purple dots cross. Okay? So this membrane is permeable to water, but not to purple dots. Now, at the start of our little thought experiment, there are not very many purple dots on the left side, and there are lots of purple dots on the right side. You'll also notice that the level of the water is the same on either side of this U-shaped tube. What that means is there's the same volume over here as there is over here. What that means is since it's the same volume, but on the left side there aren't very many purple dots, that means there are more water molecules on the left side than there are on the right. Why? Because on the right side, some of the space, more of the space, is filled up with purple dots. So there's less room for water molecules over here. So the right side here has less water than the left side. All right? And so now, since this membrane here will let water molecules cross it, over time, the water molecules will move from the left side, where there are lots of them, to the right side, where there aren't very many of them. And your U-shaped tube would look like this. We also diagram that here with the beaker, but I think it's a lot easier to see with the U-shaped tube. So that's what osmosis is, the movement of water across a membrane, a selectively permeable membrane, from where there's more water to where there's less water. So that makes osmosis just a special case of diffusion, specifically diffusion of water. There's one more kind of diffusion we need to talk about, and that is facilitated diffusion. Facilitated just means to facilitate something means to make it easier. So facilitated diffusion means making diffusion easier. Some things in a cell will be too large to just drift across the membrane or to squeeze through the membrane. They're too big, so they need a protein to help them get across. And so passive transport is the process whereby a protein allows some molecule to get across the membrane without using any energy. This is a form of passive transport. It's free. No energy required. There are two kinds of proteins that can let this happen, a channel protein or a carrier protein. Now, I mentioned those earlier, so here I also said we would figure out what that meant. So a channel protein is kind of like a tunnel, or you can think of it as a donut, right? And so it's a donut that will let something go through the membrane. Here's a little diagram of it here. So this is a channel protein, and it sits in the membrane, in the lipid bilayer, and its donut hole, its tunnel, will only let one kind of molecule through. So in this diagram, we have these black dots, and they fit perfectly through this tunnel. And so this channel protein will allow, in this case, we're saying this is a glucose molecule, will allow the glucose molecule to go from where there's more of it to where there's less of it. Remember, this is passive transport. It has to be free, so you need a concentration gradient for it to happen. So that's how a channel protein can do this. A carrier protein is the same basic idea, except it's not like a tunnel. It is what a channel protein does is bind to or grab onto its target molecule, and when it grabs onto that molecule, the carrier protein changes its shape to push the molecule to the other side of the membrane. Again, it's passive transport. It's no energy. It's moving from where there are more of something to where there are less. But the difference is a carrier protein doesn't have a tunnel or a pore through the middle of it. A channel protein does. All right? So that was the types of passive transport or free transport. And then the other kind, which is probably, equally important, is called active transport. And that's the kind that uses energy. So this is the movement of some kind of molecule across a membrane against its concentration gradient, from where there's less of the molecule to where there's more. All right, and that's kind of what we're trying to diagram. Let's look at this little diagram down here, this animation first. So here's the outside of the cell, here's the inside of the cell. Here's in this case, it's a carrier protein, and the carrier protein is grabbing onto these red hexagons and pumping them into the cell. And it uses energy to do so because you'll see there are more of these red hexagons inside the cell than outside. All right, so you're creating, or you're going against this concentration gradient. That's why it requires energy. And that's what this diagram is trying to show too. We have more of these black dots outside of the cell than we have inside. So to get them out of the cell, if you need to remove them, maybe they're a waste product like carbon dioxide, maybe you would have a pump or a carrier protein that would grab onto this molecule and shove it across the membrane from where there isn't very much of it to where there is already a whole lot. That's active transport, and it costs energy. And of course, this is a cell, so energy comes in the form of ATP, which was what we learned about in the last unit. So here's a quick summary diagram, which you also have in your notes. You may see this diagram again, so I want to make sure we walk through it quickly. Here is our lipid bilayer. Those are the two gray bars. And we have a bunch of different transport processes diagrammed in this image. So we have a bunch of different kinds of molecules that we can talk about being transported. We have big blue circles and yellow circles and small little purple circles and green squares. All right. And if you look at this part of the diagram first, you can see that there are more of all of these molecules outside than inside the cell, this hypothetical, schematic of a cell. So very few yellow dots, for example, inside, but lots of them outside. So that's a concentration gradient. All right. So here's our transported molecule. The tiny little purple dot is small, and it can just diffuse across the membrane down its concentration gradient. All right. More purple dots outside than inside. So that's an example of simple diffusion. There's also the channel, the donut or tunnel, and that's what allows the yellow dot to move across. Again, there are more yellow dots outside than inside, so the concentration gradient will drive the yellow dot into the cell through its channel protein. There's also a carrier protein for the blue circles. Still, more blue circles outside than inside, so to get the blue dot into the cell, it's free, it's passive transport, but you need this protein to help it get across. And then our final example is the green square. There are more green squares outside than inside, but if we need to get that green square out, we'll have to use some energy and pump it out using active transport and expending energy. Good? Good. All right. Some things are too large even for carrier or channel proteins to get across. Think maybe large food particles for a bacteria, or for example, your immune system has some kinds of cells that need to engulf bacteria. That's part of their part of their immune system job. So sometimes a cell has to transport something large across its membrane. How? There are two processes. One is called endocytosis. One is called exocytosis. Endo means in. Exo means out. So endocytosis is the process for a cell to get something inside itself, engulfing something. Excuse me. And exocytosis is the opposite, to spit out or to excrete or something from the inside of the cell out. So here are our two diagrams. All right, so here's an example of endocytosis. Let's say this is a white blood cell, and its job is to engulf these little bacteria here. So it forms a pit, and it pinches off the pit, and the bacteria are trapped inside, and then this pinches off to form a vesicle. So now you have moved this bacteria from outside of the cell to the inside of the cell. Could be bacteria, could be a food particle, whatever. The opposite is true over here, where we have a waste product, these little green shapes, and they need to be excreted. You need to get rid of the waste. So the vesicle in which these are stored fuses with the membrane, and you spit out the contents, so you get rid of the waste. That's exocytosis. This is endocytosis. Good? Good. All right. The last concept that we need to talk about today is the effect of concentration on cells. So cell membranes will let some things across and some things not. That's called selective permeability, right? So that can have a bad effect on a cell, or a complicated effect on a cell. For example, if you put a normal cell like this red blood cell, if you put it in really salty water, where there's a whole lot more salt outside the cell than there is inside the cell, then water is going to move into the cell and pop it. So the concentration of the solution around a cell can really be important. And in order to talk about that, we have three vocab terms: isotonic, hypotonic, and hypertonic. let's see what those mean. So hyper means more. Again, thinking back to our language of science. So hypertonic means more concentrated. So a hypertonic solution is one that has a lot of dissolved stuff compared to another solution, like the cell's cytoplasm. This is how we will usually use the term, comparing the solution that a cell is in to the cell's cytoplasm. All right. So if a cell is placed in a hypertonic solution, that means there's a lot of stuff dissolved solutes outside of the cell, and the water that's in the cell will leave to try to equalize that. And so what happens is, in a hypertonic solution, water leaves the cell, and the cell will shrivel up. Okay. Hypotonic solutions are ones that have low or maybe no concentration of a dissolved solute of some kind, maybe salt, maybe proteins, whatever, compared to the cell's cytoplasm. So there's less stuff dissolved in the solution than there is in the cell. When that happens, water will diffuse into the cell, making it swell up and maybe even pop. So kind of like a water balloon. So in this case, there's very little or maybe no dissolved stuff outside of the cell in the solution here, but inside the cell there are dissolved things, and that means that there's more water outside. Water moves from the outside of the cell to the inside, making it swell up and maybe pop. The last one is isotonic, and so iso means same. So same concentration, and that's where the concentration inside the cell and outside the cell are identical. In that case, water still moves in and out of the cell, but the same amount does, and so the cell doesn't change at all. That's what we're trying to diagram here. So in an isotonic solution, there's no bad effect on your cell. Good. Okay, here's another quick diagram of that. I think this is actually a little bit easier here. This sort of brownish region is inside of a cell, and here's the cell membrane, and this is outside the cell. We're looking at green dots. Lots of green dots inside the cell, very few dots outside the cell, and so this is a hypotonic solution. This blue part out here is hypotonic compared to the cell itself, which means that water will move into the cell. Here is an isotonic solution where we have the same number of green dots inside and outside. So water will move in and out of the cell at equilibrium, and basically at a balance, no bad effects for your cell. Hypertonic solution, lots of green dots outside, not as many green dots inside, which means water will move out of the cell and make the cell shrivel up. All right? Good. Now, that was for mammalian cells, like a red blood cell, for example, from you. That same thing also happens to plants, but they don't pop when they swell up because of their cell wall. And that's all I really want to get to with this slide. The plants have a cell wall, and it's quite stiff and rigid. And so when water is moving into a cell, it will try to swell up, but it doesn't actually pop because of the presence of the cell wall. All right. So this is, whoops, can't point, can I? This is hypotonic, this is isotonic, and here's hypertonic. All right? Good. That's it. Thank you for paying attention.